Kim grew up in Moscow and apparently met some members of our audience at a much younger age there. Obtained his PhD in biochemistry at Moscow University and established a research program there before he emigrated to the US. He's been at Northeastern University in Boston since 2001, where he's now the director of Antimicrobial Discovery Center and a university distinguished professor. So as most of you probably know, the issue of increasing resistance of bacterial pathogens to our current antibiotics is an issue that's been on the front page of newspapers, journals, and everything, and everybody's attention. And we're really fortunate to have Dr. Lewis here today to tell us about his work, because he's really been at the forefront of attacking that problem. I think I first became aware of his work and his interests when he started talking about persisters and bacterial persistence, and he's been one of the pioneers in recognizing and investigating that phenomenon, in which a small subpopulation of cells are not killed by antibiotic treatment but remain ready to grow when they get the chance in reestablishing the infection. We have a second talk from Dr. Lewis tomorrow at Lambda Lunch. That's at 11 a.m. in Building 37 tomorrow morning in the 6th floor conference room. If you need more information about where and what that is, let me know. The title of that talk is Stochastic Changes in Energy Levels Produce Drug-Tolerant Persister Cells. Today's talk, he'll be discussing his exciting work on the search for novel antibiotics, and along the way developing really interesting and important approaches to growing unculturable bacteria. And the title of the talk is New Antibiotics from the Microbial Dark Matter. And please join me in welcoming Kim Lewis to NIH.
Thank you very much, Susan, for a kind introduction, and thank you, Susan and Michael, for an invitation to this really great place. I get to see it sort of from the other side, which is also a pleasure. So what I will tell you about today is some of our basic science and how that informs our drug discovery efforts. And I will start with this sort of a textbook presentation of bacterial resistance. And what you will see here is that there are lots of different mechanisms: decreased penetration, efflux, enzymatic inactivation, or target modification. And really, each and every logical possibility has been realized in nature, and it is because of this plurality of mechanisms and their spread, at least in part, that we find ourselves in the antimicrobial resistance crisis that Susan mentioned. And apart from just the threat of infectious diseases per se, one of the things that is becoming increasingly clear, and I'm sure at the NIH that experienced a number of cases with resistant Klebsiella not so long ago, it is the fact that without effective antibiotics, we do not have the enterprise of medicine that we are used to having. So you cannot do chemotherapy or surgery if you don't have effective antibiotics. But this problem is compounded by another phenomenon, which is related to chronic infections, another way that bacteria use to avoid being killed by antibiotics. And it comes from this paradox of chronic infections. Antibiotics are ineffective against antibiotic-susceptible pathogens. Now, that makes absolutely no sense, and because it's a paradox, and that's the nature of paradox. It makes no sense. And so I was attracted to this problem a little over a decade ago. These chronic infections often associate with biofilms, and there's a long list of them, and there's, of course, tuberculosis. And one commonality among those two is the fact that the immune system is not very efficient in attacking the pathogen. And then the pathogen, which is otherwise susceptible by simple tests on a petri dish, susceptible, meaning doesn't grow in the presence of antibiotic, that same antibiotic is fairly ineffective at treating the infection in a human. So investigating biofilms of a number of species, we found this pretty simple biphasic killing phenomenon, where with increasing concentration of antibiotic, you see that the bulk of the cells are easily killed. There's nothing special about them. And then you hit upon this small subpopulation of cells that do not die. So that seems to be the culprit. This culprit are persister cells, discovered originally in 1944 by Joseph Bigger, who was unable to sterilize a culture of staph with penicillin, was very frustrated that he was unable to do that, published a paper in Lancet called these cells persisters, and that paper was largely forgotten for a couple of decades. So we, if you will, rediscovered persisters in the biofilm and realized that is very important, probably very important medically. And so we wanted to, of course, figure out what is it that makes these cells essentially invulnerable to antibiotics. Now, today I will give you some highlights of our of our work in this field. I will talk in much more detail tomorrow, and today I will give you sort of sufficient background to then explain what we're doing with drug discovery. Okay, so first of all, we wanted to know in principle what is it about these cells that allows them to stay alive, and we thought that perhaps they are dormant because they neither grow nor die in the presence of antibiotic. And so here's our view of these two phases of threat. These are resistance mechanisms, and the resistance mechanisms, all of them do essentially the same thing. They prevent antibiotic from binding to the target, and that is what allows the pathogen to grow in the presence of antibiotic. Now, in order to understand this other phenomenon, tolerance, which persisters exhibit, we need to appreciate that bactericidal antibiotics kill not by inhibiting targets, but by corrupting them. So, for example, the glycoside antibiotics will cause mistranslation. That forms misfolded toxic peptides, and that is what kills the cell, not because the ribosomes stopped turning out proteins. So if your targets are inactive in a dormant cell, or largely inactive, then the target is not corrupted and the cell survives. So this seemed to us like a reasonably good guess. We learned how to isolate persisters, got their transcriptome, and in the transcriptome we were looking for proteins, for genes and proteins that have this ability to stop important functions in the cell. And one of the first things that we noticed was the overexpression of a number of toxin antitoxin systems in E. coli. So the toxins antitoxins have been known for a while as a mechanism of plasmid maintenance. You have in a plasmid this 2-gene operon, and if the daughter cell by accident loses the plasmid, then the more labile antitoxin gets degraded. The toxin binds to its target, and depending on what that target is, either kills the cell or causes stasis. But then people started sequencing genomes and discovered toxins, antitoxins in copious numbers in genomes, like there are around 80 of them in M. tuberculosis, for example. So our simple proposition was that what these things do in the chromosome is they are persister genes, at least some of them. So here I will just give you a summary of what we and other groups, Ken Gerdes and Natalie Balabans, found over the years. What we found is that different toxins, antitoxins, will cause persister formation by inhibiting protein synthesis, degrading mRNA, or this interesting mechanism that Toby Dorr in my lab discovered, where the SOS stress response turns on this B toxin, which causes a decrease in protein force and ATP. So these are highly redundant and parallel pathways of persister formation. So armed with that information from E. coli, we then decided that it would be useful to find out what's happening in other bacteria. And at that time, Brian Conlon, a talented postdoc, joined my laboratory, and he came from a staph lab, so he wanted to figure out what's happening in gram-positive bacteria. And the first thing that he did is he knocked out toxins, antitoxins. In Staph aureus, it has only 3, unlike 80 in M. tuberculosis. And so he knocked out those 3 toxins, antitoxins, and asked the simple question: if you expose these, let's say, growing cells to ciprofloxacin, what will happen? And absolutely nothing happens. There's no difference in persister formation in the wild type or in the knockout. Same in stationary culture. So that experiment immediately told us that what we learned in the previous decade working with E. coli was completely useless. And we need to start, useless for staph, right? Not for E. coli, perhaps. And that we need to start from scratch. So we went looking for clues. And one of the clues that we thought would point us in the right direction was the fact that staph becomes highly tolerant in stationary state. So then we thought, well, maybe persisters in a growing population of staph that went into stationary early. So here's a simple test of that proposition. We have a stationary state marker cloned with GFP, and we're looking at GFP expression as the culture grows from lac into stationary state. And you see that there are stationary cells showing up in the growing culture. If you now take these cells from a growing culture and sort them out, those that are expressing the stationary marker and exposed to antibiotic, those are the only cells that survive. So those are clearly enriched in persisters. So we have this paradoxical effect happening that a growing culture produces stationary state, which I always thought that these are 2 completely different things. By definition, stationary are not growing, and yet a growing culture produces stationary cells. So what we were thinking may be happening and producing persister cells is that, of course, we know that in stationary culture, the levels of ATP are considerably lower. So we were thinking that maybe persisters are cells that have low levels of expression of key elements that are responsible for ATP production. And one of such key elements in Staph aureus is alpha-ketoglutarate, because Staph aureus likes to feed on collagen, and the pathway from collagen into central metabolism is through this portal, through alpha-ketoglutarate, which is coded, its dehydrogenase is coded by the SUCK gene. So Austin Nuxall, another postdoc in the lab, tested this and did a very simple experiment where he has a GFP reporter to SUCK promoter, and he's looking at the distribution of expression of this gene in the population. And now sorting out cells shows that those that have low levels of expression of alpha-ketoglutarate survive a hit by antibiotic considerably better than the wild type. So this is our first mechanism. We were pretty happy about it because the alternative was that there is no mechanism because lots of things are going to affect the levels of ATP. But now we know that there are some key elements that do that, and tomorrow I'll tell you about some additional mechanisms in E. coli that we found. Okay, so here's our additional view of what's happening with persister formation. I think this is a general mechanism. The mechanisms that we and others have been studying before are toxin, antitoxin dependent, and they are sort of specific and perhaps selective for given bacteria. This, I think, is a general and important mechanism, maybe the most important mechanism, and that is that all of the antibiotic targets require ATP, of course. And so if ATP is present at normal levels, then you get corruption of these targets by these antibiotics and death. And then if stochastically the level of the target decreases, you get dormant cells because ATP went low, and you get the target shut down. So that very simply explains the nature of persisters. It's a pretty simple explanation. I must say that we overlooked it. We could have discovered this 10 years ago, but we didn't for reasons that I don't understand. Okay. So apart from that, apart from the basic biology of persisters, there's another thing that I wanted to tell you about, and that is the important question of the clinical relevance of persisters. So a friend of mine, Lou Rice, who's head of medicine at Brown, he told me a number of years ago that it is very interesting that what you guys are doing with persister cells, and I'm sure amusing, but it has nothing to do with clinical manifestation of disease. We know why chronic disease is difficult to treat is because pathogen hides somewhere so that antibiotics don't reach it, and that's all there is to it. So it's difficult to address that question. We were thinking to do a Koch postulate style experiment where you isolate persisters, which we learn how to do. We could introduce them into an animal and see if that animal becomes tolerant to killing by antibiotics. But the moment you introduce persisters into an animal, they wake up. So you cannot do Koch postulates on persisters. So that was quite a big problem for us and for others in the field. And the clue to solving it came from an experiment that we were doing on a completely different subject. So a grad student in my lab, Pooja Balani, was trying to catalog all potential persister genes in E. coli. And the way she did that was to perform this pretty simple selection experiment where you hit a culture with antibiotic, collect surviving persisters, repeat it a couple of times. And then you get enrichment and mutants that make more persisters. And now you can sequence the genomes of these cells and figure out where those potential persister genes are. But if you look at this experiment, what you realize is this is exactly what happens when we people are treated with antibiotics. We get periodic high doses of antibiotics, and if being persister is useful for the pathogen, then there will be selection for high persister mutants in the course of antibiotic treatment. So in a way, millions of people participated in this experiment, including probably everyone present in this audience. And so we simply need to look at the results of this experiment and decide whether there are high persister mutants among clinical isolates. So that experiment we did, but first I will show you what we found in the in vitro experiment. In the in vitro experiment, most of the high persister mutants turn out to be mutants in different positions of this hip-A toxin. So these are gain-of-function mutations in hip-A toxin, which produce 100 to 1,000 times more persisters as compared to the wild type. So an enormously strong phenotype, which of course gives the pathogen considerable advantage in survival. So then Pooja screened a large collection of E. coli isolates, primarily from patients from relapsing urinary tract infection. It's a chronic condition, and found that in half of these strains there were mutations in the hip-A locus, including those very mutations that we found in vitro. So of course we wanted to understand mechanistically what is happening with this, and we knew something already about the hip-A protein. So what we knew initially came from a study that we did a number of years ago. We were trying to understand what is it that hip-A does in the cell to stop it from growing, and we didn't have any leads, and hip-A was not homologous to anything according to blast searches. At which point I gave a call to Eugene Kunin, and I asked him to take a look at hip-A, which doesn't look like anything in the database. So Eugene reluctantly agreed to do that, after some arm twisting. And in a couple of days, Kira Makarova and Eugene sent me an email which said that HIP-A was a kinase related to the TOR family of kinases, which I thought was nonsense because there are no TOR kinases in bacteria. But the active sites lined up very well, and we found that indeed HIP-A is a protein kinase. And that was a sort of, in my mind, an example of pure intelligence informing science, right? That was a good clue. So HIP-A is a protein kinase that phosphorylates glutamyl tRNA synthase and stops translation. But normally in the cell, it is present in the form of a dimer. This comes from our collaboration with Dick Brennan and Marie Schumacher crystallography team. And so normally, HIP-A is largely inactive. All the mutations that we found, the gain-of-function mutations, they happen at the interface between these dimers, opening up the pathway from ATP to the active site and activating the kinase. That's why you get considerably more persisters in these mutants. So this is the case when we understand mechanistically the nature of this high tolerance mutation, and we by then assay the number of other pathogens. So all pathogens we looked at, and these are M. tuberculosis, Pseudomonas aeruginosa from cystic fibrosis patients, and Staph aureus. In all cases, we find high persister mutants in clinical isolates. In cystic fibrosis, about half of isolates we find are high persister mutants. So it looks like there are these two phenomena. One is heritable resistance, and the other one we call heritable tolerance. And to make matters even more difficult for us, there was a paper by Natalie Balaban's group that just came out in Science that showed that in the course of antibiotic treatment, pathogen first acquires tolerance mutations that help the pathogen survive high levels of antibiotics, and on that basis, resistance mutations are then acquired, right? So these two mechanistically distinct ways of protecting themselves from antibiotics, they collaborate, if you will, in producing resistant mutants. So we need to worry about both of these phenomena and take care of both of them. So what is it that we learned? How does that inform our drug discovery efforts? So the pathways of persister formation in the model E. coli are multiple and redundant. And many of these persisters are going to be low in ATP. So if you have redundant pathways, that means you don't have a target. If you have low ATP, that means that no antibiotic is going to kill. So what we found tells you very simply that drug discovery is rather hopeless. So from that unpromising basis, we were thinking how to get something that's going to kill persisters. When Mother Nature comes up with a perfect defense, what we know is that it also provides a response to that perfect defense. And so we were wondering if among natural products there may be something that evolved to kill persisters. Because producers of antibiotics face a very similar problem to us. They want to kill their neighbors, including their persisters. So what we're looking for a compound that will bind an important target in a dormant cell, corrupt it, and kill the cell, and that is not going to require any ATP. So that's the kind of a magical compound we are looking for. But once you formulate that, then indeed there is a compound that comes to mind. This is acyl depsipeptide, a natural compound made by Streptomyces hawaiiensis and discovered by a group from Eli Lilly. Apparently someone from Eli was vacationing in Hawaii and brought back this strain that produces this antibiotic. So Lilly had a look at this compound, and at the time, this was 1985, everybody was looking for broad-spectrum antibiotics. This compound only kills gram-positive bacteria, so Lilly dropped it. About 20 years later, we were experiencing an epidemic of MRSA, which is still ongoing, and other gram-positive bacteria have become highly problematic, drug-resistant ones. And so a group from Bayer took another look at this compound. They made a more active derivative, ADEP4, and they also dropped the compound, but before dropping it, they determined the principal mechanism of action. It is very interesting, and that's what attracted our attention. So, ADEP attacks the CLIP protease. The CLIP protease will normally recognize misfolded peptides with the aid of this ATP-dependent chaperone and digest them. In the presence of ADEP, ATP is no longer needed. So the pore of the protease stays open, and it can degrade misfolded peptides. So that's the key, of course. That's exactly what we're looking for. We're looking for a target that can be corrupted without any requirement for ATP. So that seemed to us like a perfect candidate for an anti-persister compound. But there was a problem, actually two problems. These two papers. One of them reported that in the presence of ADEP, CLIP only digests peptides that are getting off of the ribosome and did not yet have a chance to fold properly. So that will, of course, only happen in a properly growing cell, not in a dormant cell. And the other paper identified the primary target, FTSZ. FTSZ forms the septation ring, and of course, that is also only required in dividing cells, not in dormant cells. So the question is, what do you do when facts clearly contradict your beautiful hypothesis? You have two basic options. One is you decide that your hypothesis is wrong, and the other, you decide that facts are wrong. So I took a very careful look at these two papers, and I didn't find any problem with them. They're very well-executed studies. But like most biochemistry studies, they are performed on a fairly short time span of about 10 to 15 minutes. And that is an issue because antibiotics act at a time frame of hours and days, not a time frame of minutes. So we decided to repeat this experiment at a more realistic time frame of 24 hours with non-growing stationary state E. coli. This is work of Brian Conlon, who's now at Chapel Hill. And so here is the full proteome of Staph aureus after treatment with ADEP, and you see massive degradation of about 400 proteins. So it looks like ADEP forces the cell to self-digest. And of course, we had a look at what happens with killing. So this is a control with ciprofloxacin. These are growing Staph aureus. You see a biphasic killing. Same happens with rifampicin. If you add ADEP, you get complete sterilization. That was the first time that we saw complete sterilization with anything which is not bleach. That was encouraging, and then we recalled why Bayer dropped the compound. They had a reason to do that. CLIP-P protease is non-essential, at least in vitro. So you get high probability resistant mutants that arise when there's a null mutation in the CLIP protease. The CLIP mutants become completely resistant to ADEP. But we decided, since ADEP has this potential to sterilize, we'll simply combine it with another antibiotic, doesn't matter which, to prevent the rise of resistant mutants, and we'll use a combination. And so here you see sort of an attempt to kill an in vitro biofilm with conventional antibiotics, which is not doing very well. And here's a combination of ADEP with rifampicin, which completely sterilizes a biofilm. And we also had a look in vitro. AstraZeneca developed this model, developed an in vivo model of a biofilm infection in the mouse of a neutropenic mouse thigh. And here you see the growth of a massive biofilm, and this is an incurable infection. And indeed, regular antibiotics do very little in this model. They kill growing cells, but then you hit upon the non-growing persisters, and this is an incurable infection. And again, ADEP rifampicin sterilizes in this infection. So that gives us an example of an anti-persister compound that tells us that nature did evolve anti-persister compounds. That was the first, but I'm sure not the last anti-persister compound. And a biotech company is now developing this into a drug. Okay, so in the remaining time, I will tell you about our efforts to discover new antibiotics. And I will start with this brief introduction into where the field is at the moment. So the field started with the golden era when the main classes of antibiotics were discovered, and they were discovered primarily from soil actinomycetes. And this was based on an extremely simple screening that Selman Waxman developed and got the Nobel Prize for the screen. And the screen essentially emulates the accidental discovery made by Fleming. When Fleming found that if he had a plate with Staph aureus that he was growing, and then he got an accidental contamination, there was a zone of inhibition around that contaminant. In the case of Fleming, the contaminant was a penicillium, which was making penicillin, and Selman Waxman realized that you can use that as a systematic screen, and he had sort of the genius to hit upon the world's best producers of antibiotics, which are the streptomyces. How he managed that, I don't know, but he did. And so this very simple screen produces the major classes of antibiotics, and that goes on for a while very successfully, and then something happens around the 1960s. The mechanism of discovery is turned off. We go into the dark ages, and then there's a bit of a resurgence of activity, but this resurgence primarily defaults back to the golden era, meaning that we are primarily taking initially failed leads and trying to resuscitate them because we don't have novel compounds. So we have a pretty good understanding what happened around 1960, and that is that this resource, the soil microorganisms, was in a way largely overmined by screening because only 1% of them will grow on our petri dishes. The rest are the microbial dark matter that doesn't grow. So with my colleague Slava Epstein at Northeastern, we decided that we will try to revive the Waxman platform by going after this microbial dark matter. So this is a simple experiment that sometimes is performed in our undergraduate laboratory, which illustrates what uncultured bacteria are. You take a sample from the environment, like marine sediment or soil, and you put one droplet under the microscope and you count the number of cells. The other droplet goes on a petri dish, and you try to figure out how many colonies these cells produce. And the difference in the counts between the number of cells and the number of colonies that they produce is known as the Great Plate Count anomaly. That is perhaps the oldest problem described in microbiology. It was discovered by Winterberg, an Austrian microbiologist, in 1898. Since Winterberg, there's been a considerable effort to tinker with the media on the petri dish to try to push these numbers up, and that did not work. So we decided to do something different. Instead of growing bacteria on a petri dish, grow them where we knew for a fact that they do grow, and that's their natural environment. So we needed to come up with a gadget where bacteria can grow in pure culture in their natural environment. So here you take a sample from marine sediment, dilute it, and place between these two semi-permeable membranes that get glued on an O-ring, which we buy at Home Depot, and this contraption known as a diffusion chamber goes back into the environment. So now everything diffuses through the chamber, and essentially you're tricking bacteria. They don't know that they're not in their natural environment, and not surprisingly, lots of stuff grows. we get recovery close to 50% in the diffusion chamber as compared to the petri dish from this environment, which is really low in cultivable bacteria. So our latest iteration, which is developed primarily in Slava's lab, the iChip, is a massively parallel version of the diffusion chamber. It's just a piece of plastic with lots of little holes, which you dip in a suspension of bacteria. Each well captures approximately one cell. So then you get isolation and growth in one step. You cover this with semi-permeable membranes, and this goes back into the environment with incubation. And you get microcolonies in these wells. So, of course, we were very interested in trying to figure out why uncultured bacteria do not grow in a petri dish. What is, if you will, the molecular mechanism of uncultivability? So this is work from my lab, and we decided to start with this, with the beach sand as a model, where I suspected that on these particles of sand will be biofilm communities of microorganisms. So this is what you actually walk on when you walk on the beach. You walk on this. This is a, I don't know what this is, but it looks very imposing. So then we had a simple hypothesis that perhaps uncultured bacteria, at least some of them, do not grow because they require growth factors from their neighbors. So here's a test of this. You dump a heavy inoculum from that sand particle on a petri dish, and you get a whole zoo of colonies growing. And then you say, well, maybe this small colony grew because it happened to be in the vicinity of this bigger one that was releasing a growth factor. So here's a test of that idea. Here's a sort of a simple test where you patch one of them, the bigger one, and it grows as a colony, and the other one you spread on a petri dish, and it only grows around this bigger one. That, first of all, tells you that the idea is correct in principle, and that you have a bioassay. You can purify the growth factor by simply growing up the cultivable bacteria and fractionating it. So that we did, and what we got is these growth factors, which turn out to be siderophores, iron chelators. And if you drop one of them on a petri dish, it recapitulates the effect of the helping organism. So siderophores are iron chelators, and normally in an aerobic environment, iron exists in the form of insoluble iron 3. So bacteria release the siderophore and then take the iron back and reduce it. Uncultured bacteria, interestingly, lost the ability to make their own siderophores. They come from unrelated taxons, so it's a secondary loss. Why they lose their ability to make siderophores, and with it they lose their liberty, they always depend on someone, that is a fascinating question. But now they have to steal siderophores from their neighbors, and that's how they grow. So we got very excited when we found this because I thought that we'll just keep finding additional growth factors and we'll close the gap in the great plate count anomaly. That did not happen. With siderophores, we can explain approximately 10% of uncultivability. We can go from about 1% to 10% cultivability. The next growth factor gave us 1% and then nothing. So I don't understand at the moment why 90% of uncultured bacteria do not grow, but I have a postdoc in my lab who is supposed to find an answer to this question by the end of this year. So one useful thing that we found is that once grown in a diffusion chamber, especially if you re-inoculate from diffusion chamber to diffusion chamber, with high probability you get growth on a regular petri dish. So that, of course, gives you now access to considerable biomass and antibiotic discovery. And we've been collaborating with this startup, Novabiotic, and so far we have 29 new compounds. So I'll tell you about 2 of them. So one compound came from an effort to solve a very tough problem in natural product discovery, and that's the enormous background of known compounds and junk. And so the way chemists try to solve that problem is to ask the question whether you have a new molecule in your extract. That takes time and energy, and the answer is not entirely satisfactory because your new molecule can be a new detergent, which is not terribly exciting. So we decided that one way to solve that problem is if you go after species or group selective compounds. So nature has very few, if any, compounds that have been known to hit a particular group of bacteria. I don't mean gram-positive. It's a very narrow group, like mycobacteria, for example. So then, if nature makes compounds that are specific against mycobacteria, then you can do a very simple thing. You take an extract, and you're looking for this property. You're looking for inhibitory activity against mycobacteria, but not against, for example, Staph aureus. If you can do that, then you can immediately say that your extract probably contains a new compound active against mycobacteria. So by doing that, we discovered this new antibiotic, Lassomycin, which is very nicely selective against mycobacteria. It doesn't hit bacteria from the human microbiome. And it has an interesting property. If you look at stationary MTB, it's very tough to kill them. This is our best killing compound. Rifampicin gives you biphasic killing with surviving persisters. With Lassomycin, we don't see any indication of surviving persisters. So we got very interested in this compound, and the postdoc, Katya Gavrish, got resistant mutants for Lassomycin, and all the mutations mapped into, well, not mapped, we don't do mapping anymore, but sequencing showed that they all fall into the same locus. And that is the CLIP-C1 subunit of the mycobacterial CLIP-P1, P2, C1 protease, which is fairly specific for mycobacteria. It's also essential in mycobacteria, unlike in staph. So Lassomycin, which looks like this, that's why we gave it the name Lassomycin, looks like a lasso. It binds the CLIP-C1 subunit, and it does two different things. It inhibits in vitro the protease activity completely, but then also there's strong activation of ATPase activity. So if you look at what happens in whole cells, Lassomycin collapses ATP in whole cells, and that is apparently what contributes, or maybe is primarily responsible for killing by antibiotics. What I think is happening is that there is this mechanism of persister formation that I just told you about, which is ATP depletion, but not to zero. And it looks like Lassomycin pushes the system down sort of an extra step into the abyss of irreversible dormancy. I think it's a very sneaky mechanism, sort of piggybacking on the natural way to make persisters to get rid of them. So it looks like we hit upon an Achilles heel of persisters, and it is this particular molecular machine which is under attack, the CLIP protease. So in bacteria like Staph aureus, ADEP forces the protease part of this machine to degrade proteins, and that's how it kills dormant cells without any requirement for ATP. And in mycobacteria, a very different compound hits a different part of the same machine, forcing it to digest ATP. And of course, that doesn't require ATP either. It just mops up whatever ATP is in the cell. Why this particular machine is under attack by completely unrelated natural products is a fascinating question. Okay, so while I'm sure that there are other anti-persister compounds to be found, I will tell you about another compound that we found sort of related to this general problem of antibiotic resistance. And this is Teixobactin. So this was discovered from this very interesting uncultured, and unusual uncultured bacteria, Eleftheria terra. This is a beta proteobacterium, and that particular group of bacteria is not known to make antibiotics. It is a gram-negative bacteria, very different from your usual actinomycetes. it forms a new genus, and we had to give it a name, so we gave it this name, Eleftheria terra, which is free earth. You'll see in a moment why we gave it this name. This name is a mixture of Greek and Latin, which you're not supposed to do in taxonomy when giving names to creatures. But I figure that nobody cares about these things anymore, so this is the name of the creature. Teixobactin is an interesting complex molecule, and we checked it out for activity and found that it hits a long list of gram-positive bacteria, including some nasty pathogens like MRSA, enterococci, for which we have very little options clinically, M. tuberculosis, and not so much against gram-negative bacteria. We tried to get resistant mutants to this compound and failed, which was very frustrating because, first of all, you do not have an easy path to determine the mechanism of action, but more importantly, in our experience, if you have no resistance development, that means that you discovered a new detergent. But in parallel, we were also running a cytotoxicity assay. So this is against human cells. There's no activity against human cells. So that was very interesting. This is the first time we saw a compound where there's no resistance development and no cytotoxicity. We examined it in a number of animal models. I'm not going to go over this in any detail, just to tell you that it is pretty efficacious in several simple models like septicemia, protection, mouse thigh infection, and mouse lung in pneumonia. This is all mouse. So that gave us some additional encouragement that this compound is worthy of investigating. And so then we took a very in parallel, the team at Biotic and in my lab, we decided we'll in parallel investigate resistance development. And in microbiology, there's a protocol. If you have a problem getting a resistant mutant, there's a protocol how to do it correctly. So we did it correctly. Here's the control with ofloxacin. You add antibiotic at sub-inhibitory concentrations, and each day you up the concentration. So if you have a mutant with any increase in resistance, even minimal, it will take over the population. You encourage resistance development. And then after about a month, you're supposed to do it for about a month, you get an enormous increase in resistance, and this is pretty much how all antibiotics in current clinical practice behave. With Teixobactin, we did not see any changes. So that was very surprising. Now, you performed a specificity test to see if this is a compound hits a specific target. The test is very simple. You simply follow the rate of label incorporation to these major polymers, and in this experiment, Teixobactin clearly selectively inhibits peptidoglycan synthesis and nothing else. So we know now that this is a cell wall acting inhibitor. And there's no resistance development to it, so we concluded that this compound does not have a protein target. Because proteins will always mutate. You will always have a mutant protein that does not bind your antibiotic. A realistic target, which is not protein, is a precursor of peptidoglycan, lipid 2. So we started with a simple test. We had growing Staph aureus, which are not growing in the presence of Teixobactin. If you add lipid 2 to the test tube, you restore growth, which means that Teixobactin is sequestered by lipid 2. So we're collaborating with Tanya Schneider, who performed this, a simple biochemistry experiment where you can isolate lipid 2 and see it on thin layer chromatography. But as you add Teixobactin, you can no longer extract lipid 2 from solution, telling you that it is sequestered. And so using that method and adding different analogs or versions of lipid 2, you can then zero in on the exact moiety of lipid 2 that Teixobactin binds to. And here's this moiety. It is the lipid pyrophosphate sugar part of the peptidoglycan precursor. This is the growing peptidoglycan. This is what Teixobactin binds to. The nature of the sugar is not important, so that told us that it will also probably bind, and indeed it does, to the precursor of another important cell wall polymer, teichoic acid. It has the same structure, just a different sugar. So this investigation tells us an interesting thing. It indicates that nature evolved the compound to be fairly free of resistance. It hits two targets. Neither of these targets is mutable, right? These things are not coded by genes, at least not directly. The target itself is exposed on the surface of the cell, so you cannot pump it out. It's already outside. It appears that the main dogma that we've been operating under, which is that bacteria will develop resistance to everything, and in many cases rapidly, is probably incorrect. They're probably compounds like Teixobactin that evolved to be largely free of resistance development. So going after those compounds, I think, makes a lot of sense. So with that, I will thank my team. I will thank my collaborators and support, especially the NIH, which has been supporting the work of my team over the years. And I thank you for listening. Yeah, there's a question there, but I think you need to go to the microphone. Yeah.
Yeah, well, you were speaking about biofilms and that you had kind of beaten them. You didn't say anything about the structure of biofilms. Supposedly they have an outer layer of protectors, and they have an inner layer of guys that are kind of protected, and then there's a lower layer or something else. There's certain hypotheses of how they are. Can the type of thing you're talking about actually beat all the layers of a biofilm, or just particular bacteria that are laid out flat before a biofilm, an actual biofilm formation? That is significant, because what you're talking about is medical devices that are going to be indwelling, and they do develop all those different layers of things. So.
Sure. Yeah. So I think they're related to, this is of course an important question, and there are two aspects to it. One is the ability of the compound to penetrate through the biofilm, and the other is the ability of the compound to kill cells in the biofilm. So the ability of compounds to penetrate into the biofilm has been well demonstrated, with few exceptions. Like most compounds penetrate fairly well into biofilms, like fluoroquinolones or beta-lactams. There's some problem with aminoglycosides. There's some retarded penetration. The main problem with the biofilm is not penetration into, but the ability to kill cells in the biofilm, which are dormant, and they are first and foremost dormant persisters. What we showed previously is the most difficult cells to kill are not cells in the biofilm, but cells in a simple stationary culture, right? Because the biofilm is growing, and the stationary culture is not growing at all. So ADEP kills cells in stationary culture and in vitro biofilms and in vivo biofilms that we have tested.
Thank you.
So, Kim, in the first part of your lecture, you spoke about different kinds of mechanisms behind persister formation. You kind of pointed out that there is a very general, non-specific mechanism that involves more or less spontaneous fluctuation in ATP content, and then there are specific mechanisms brought about by toxins and antitoxins. So could you elaborate on this somehow? Under what kinds of circumstances, in what situations these specific mechanisms are involved?
Well, I will elaborate, Eugene, but my elaboration is limited by my ignorance. So I will only talk about some sort of clear examples that we know that we have at the moment. So a good example that we have is the induction of TSB toxin. So during DNA damage, you have activation of the SOS response and, of course, DNA repair that response activates. And you have also an activation of expression of the TSB toxin, which is regulated by the same LEX repressor, which controls the entire SOS response. So TSB gets activated, inserts itself into the membrane. It is a typical antimicrobial peptide, which is a very strange molecule. that diminishes the protein motor force, decreases ATP, and that's how you get persisters under those very specific conditions of DNA damage.
So in other words, under particular types of stress, you would implicate toxins, antitoxins, but under general conditions, the spontaneous generation of persisters will be sort of stochastic fluctuation.
I think, yes. My feeling is that probably under conditions where the culture is not necessarily stressed, is primarily guided by fluctuation in ATP levels.
Okay, that does make sense. Thank you.
Let me ask two questions about the sort of environmental microbiology that comes out of some of the things you're doing. So for instance, for the siderophores that you see being borrowed by what was otherwise non-culturable ones, are those going to be specific to who's in the neighborhood? If you tried an unculturable from someplace else, would it not use those? Is there a specificity?
Yeah, that's a great question, and we examined that, and what we found is that there's every logically possible scenario, right? So we found some bacteria that will only take a siderophore from a particular neighbor and no other siderophore. And then we found those that we could induce growth with each and every siderophore that we could buy commercially, and everything in between, right?
So we'll eventually understand it,
yeah.
But not yet. And probably the answer to the other question. So the bacteria that gave you your antibiotic, that hits the peptidoglycan, anything known about why it would be making this and presumably killing off all its gram-positive neighbors, or?
That's an excellent question. So, well, I mean, in one simple scenario, you want to get rid of your neighbors, right? That's just a common sentiment of, at least of wild creatures.
We like our neighbors, but that's
right?
a special case.
But there is one particular property of Teixobactin that kind of tells us why that compound. So what we found about Teixobactin is that it has a terrific activity to lyse cells, considerably better than any other cell wall-acting antibiotic we looked at, probably because it hits these two targets. And so I think that this creature, Eleftheria terra, is a hunter. I think it hunts gram-positive bacteria, lyses them, and eats them with the help of this compound.
Can there be drawn a parallel between the mechanism you proposed and the tachyphylaxis, which occur when, say, a receptor stops to respond to repeated applications of an agonist, which is also ATP dependent?
Why not?